Hello guys, how are you doing? This is Mr. Marashani. Welcome to our online tutorials. Okay, today we are going to talk about reproduction in plants. Patakataura ne about research work, takataura about reproduction in human. But today we are going to talk about reproduction in plants. Reproduction in plants. I want you to listen very carefully because this is our last topic in combined science under biology section. And after this, we're going to write a big topic on biology section only. Then after that, we're going to move on and talk about chemistry section. Okay, so I want you to listen because some of the things that I'm going to teach you here are the things that are in the examination that I'm going to give you tomorrow or a day after tomorrow. Okay, so this is chapter 6 of combined science in biology section, which is reproduction in plants. What is reproduction in plants? We're going to talk about sexual reproduction, and we're also going to talk about asexual reproduction. Okay, sexual reproduction is reproduction through seed. That is sexual reproduction. Is the reproduction through seed? Because we are talking about reproduction in plants. Tiri kuti kudeveloper kweplant nyowani tichishandisa seeds. That is reproduction through seeds. While asexual reproduction is the reproduction without seeds. Reproduction without seeds. Asexual reproduction tikutaura zvinhu zvakaita sechibage, zvakaita sebeans, handitika. Then asexual reproduction tikutaura reproduction without using seeds. Yatinogona kupa zita rekuti vegetative reproduction. In that vegetative reproduction, we are talking about cassava, anonzi madumbe. We are talking about sugar cane. Kuti hatishandise seed, but tinotoona pachitika inonzi iyo reproduction. Okay, so in detail I'm going to talk about sexual reproduction, as you can see. Okay, the first statement there, it says sexual reproduction is made possible by the flowers. Is made possible by the flowers. So I am going to teach you about this structure of flowers. We have two types of flowers. Number one, it's insect pollinated flower, and number two, it's wind pollinated flower. So in other words, for you to know what we are talking about when you are talking about wind pollinated flower and insect pollinated flower, you need to know what is pollination. All right? Pollination is just the transfer of pollen grains from the anther to the stigma of a flower. Transfer of pollen grains from the anther to the stigma of a flower. Okay? Ma pollen grains anoproduced ne part of a flower inonzi iyo anther. So kuzofamba kwema pollen grains from this anther achienda ku this part inonzi iyo stigma. That transfer, that movement is called pollination. Is called pollination. Let me repeat this. Pollination is just the transfer of pollen grains from the anther to the stigma of a flower, right? Ndikatinini ma pollen grains, they are being produced at the anther. Then the movement of pollen grains produced from the anther to the stigma, and that movement or that transfer is called pollination. So as you can see now, you are beginning to getting the reason why it is called insect pollinated flower. I think this one is called insect pollinated flower. Why? Because the agent of pollination, which means the thing that carries pollen grains from the anther kuendesa ku stigma inenge iri insect. And this one is called wind pollinated flower. Why? Because the agent of pollination that carries pollen grains from the anther to the stigma is wind. Okay, so already I am giving you the difference between insect pollinated flower and wind pollinated flower. The first difference is that the agent of pollination for insect pollinated flower is insect, and the agent for wind pollinated flower is wind. That is the first difference. Okay, so let me give you the functions of parts of the flower. we have this petal. Ma petals ese a insect pollinated flower anenge ari ma bright petals. What is the purpose of them being bright? So that they can attract insects. Right? There is this stigma. The purpose of stigma is to receive pollen grains. There is this anther. The purpose of the anther is to produce pollen grains. Right? The filament is to support or to hold the anther. Then the style is there to hold the stigma or to support the stigma. Then the purpose of this ovule is to develop into seed. After fertilization. And the purpose of this ovary is to develop into fruits after fertilization. The nectar, it is food for insects. I think these are important parts of a flower that you need to know their function. Okay? There is also what we call, as you can see here, the stamen is the male part of the flower, is made up of the anther and the filament. Do you know the stamen? The stamen. Saka vanogona kubvunza kuti what is the name of anther and stigma? Inonzi iyo stamen. Tikabatanidza anther and stigma ndo inotipa inonzi iyo stamen. Right? Koita inonzi a carpel is the female part of the flower made up of one stigma, style, and ovary. Those no gadzira inonzi iyo carpel. Okay? Right? Ma pollen grains I think wamaona ndo atati anoproduced ne anther. Apo ndo pakanzi ma pollen grains are the male sex cells of the plant. Pollen is found on the anther. Ma ovules are the female sex cells of the plant. They are located in the what? In the ovary, which means inside the ovary, as you can see up. Those are zakatoita. My ovules are inside the ovary. Okay? Right? Pollination already defined that transfer of pollen grains from the anther to the stigma. But there are two types of what you call pollination. Number one, self-pollination. What is self-pollination? Is when pollen grains are transferred from the anther to the stigma of the same flower. One flower. My pollen grains are from the anther achienda to the what? To the stigma, but within one flower. Don't self-pollination. Which means cross-pollination. There's a good panapa iri flower. Then my anthers approach my pollen grains, but my pollen grains are move achieve this flower achinda kune another flower. So a transfer of pollen grains from one flower to another flower of the same type. Though cross pollination, but self pollination what is repeated kappa same flower. Cross is between two flowers but of the same kind. Kana chiri chibage chino no share ma pollen grains kune chibage. Kana rimwe plant rino share ma pollen grains acho to the anther of another plant, the same type. Anditika? Right? Let's go. We want to talk about the differences between insect pollinated flower and wind pollinated flower. Okay? Right, my insect pollinated flower, they are brightly colored petals. Because my petals of insect pollinated flower, they are brightly what? Colored white to attract insects. But a wind anenge are dull petals. Why? Because its agent of pollination is wind, not insect. Okay? Then you have pleasant smells, scented, cannot pleasant what? Smells. Ndo attract ma insect, but ukakuna boza hapana need ku attract ma insects, but its agent of pollination is wind. Okay? Then my insect pollinated flower, they produce sweet nectar, which is food for what? For insects. But my wind do not produce sweet what? Nectar. Why? Because its agent of pollination is wind, not insect. My anthers are inside the flower at this one. Then my anthers are exposed. You can check my diagrams. Stigma is smaller and inside the flower, then exposed bush stigma. Yes. Then my pollen is thick, larger and fewer. My pollen is light, abundant. Why are they light? For them to be easily blown away by wind. That's it. On the way. Right? We want also to talk about germination. What is germination? Germination is whatever development of when a seed develops into a new plant. Let me say that. When a seed develops into a new plant, do inonzi iochi germination. So there are conditions necessary for germination. Yes, vakatipa. Right, there are conditions are necessary for germination. Tine inonzi oxygen, tine moisture, tine inonzi a suitable temperature. Why teach a suitable temperature? We can't say temperature because temperature is not enough. It can be low temperature which is not suitable. It can be high temperature which is not suitable for a specific seed. So in a suitable temperature for that seed to germinate or to develop into a new plant. Okay? These are the conditions necessary for germination. In other words, we are referring to the conditions that facilitate or promote the process of germination to take place. Panofanirwa kunge pane zvinhu 3, which is oxygen, warmth, and a suitable temperature. Okay? Right? Let's move on to this experiment yakataurwa panapa. As you can see here, this is the experiment of at low temperature germination will not take place. Why? Because my enzymes would be inactive. My enzymes anoita kuti seed regerminate anenge ari inactive, anenge asiri active, zvokonzeresa kuti seed risa germinate or seed risa developer into a new plant. So as you can see, we are already talking about the importance of having a suitable temperature. We must have what we call a suitable temperature for germination to take place. Not temperature, because temperature it can be low. Kanayava low, it causes enzymes to be inactive. It causes enzymes to be inactive. And this statement says they require an optimum temperature. Optimum, it means suitable. They require an optimum temperature in order to work effectively. What is it to work effectively? In other words, we are referring to activating the enzymes so that they can promote the process of germination. Okay? Without oxygen, without moisture or water, you can say moisture or water. Without oxygen, without moisture or water, germination does not take place as well. Why? Because metabolic reactions require what? Water. Metabolic reactions require water, and those reactions, they are very, very important. Why? Because they promote the process of germination. And also oxygen tinoida nemakayei, because it is required for the process of respiration. Takatola ma seeds by experiment ataita, I think taita experiment of on what? Can't remember. But pan experiment yatakaita, yeah, it's on respiration. Yes, it's on respiration. Yatakatora ma seeds, tikatotora 30 seeds. Watora maseeds ako wama divide into 2, watora 15 uku, watora 15 uku. Maseeds ako wama some test tube titoa testa kuti ma germinating seed ano produce heat energy here. Ndoba mama acho tama soka tama some boiled water for 5 minutes. Ipapo taida kuuraya ma cells kuti asazo germinate. Tida testa ano produce heat energy here. Kuda kuti ma germinating seeds ano produce heat energy. Germinating seeds, they release heat energy. So this oxygen is needed by seeds so that they can germinate, so that they can release energy, which is necessary for the process of germination. Okay? Pana patakapuwa ma test tube 4. Now tunyasoona. Remember, this is our last lesson on biology section. Okay? Right, test tube number 1 zerine ma seeds, moist cotton wool, liquid absorbing oxygen. ndo test tube rectangle, right? Then koita test tube rech 2 riri kunzi iro rine masidi zvekare, rine liquid absorbing carbon dioxide. Koita liquid number, test tube number 3 rine water, harina liquid inoabsorb oxygen kana carbon dioxide, but temperature inongori 15. Then the last one, the test tube 4 rine water, temperature yacho iri low, rine 1 degree Celsius, masidzi arimo zvekare. Right, izvo ndo zvatinoona matest tube. Pari kunzi patest tube 1 germination will not take place due to lack of oxygen. Why paita lack of oxygen? Because mune this liquid ish iri kuabsorba oxygen, saka yakona kuitika seeds akona kugerminate. Why? Because there is a lack of oxygen, since this liquid is absorbing oxygen, and that liquid is called pyrogallic acid. That liquid is called pyrogallic acid, which is used to absorb oxygen. That is the reason why in test tube 1 there is no germination that is going to take place. In test tube number 2, germination will take place. Why? Because carbon dioxide is not necessary for the process of germination, of which the liquid that is in test tube 2 is the liquid that is absorbing carbon dioxide, not oxygen, which means in this test tube there is abundance of oxygen, which promote the process of germination through what you call respiration. That's why it's in test tube 2 munoitika respiration, because liquid irimo is not absorbing oxygen, is absorbing carbon dioxide, which is not necessary for the process of germination. Right? In test tube 3 is the control. Why? Because germination will take place. Mune liquid, which is water, mune temperature is suitable, which is 15 degrees Celsius, mune seed, mune oxygen, mune zvese zviripo, so germination will take place. Then test tube 4, germination will not take place. Why? Because the temperature is too low. Tataura kumusoro kuti temperature kana iri too low chinoitika, it will cause enzymes to be inactive. It will cause enzymes to be inactive. Okay? Right? Let's go. Germinating seeds respire, provide respiration, provide the energy needed for germination and growth. There are mainly two types of seeds, anonzioma monocotyledon and dicotyledon. Regarding taurende zao, then to vatapedza a lesson, right? Panenonzi dicotyledon seed, example ndoye bean seed. Apa tisatatidzwa external ne internal structure kunze nemukati. Anditi, right? This one though the external and this one is the internal, but yes, bean seed. Okay, so tichangotaura ma functions of ma parts atiri kuona bean seed. Pane inonzi iyo micropyle kana kuti micropyle. This one micropyle inoita basa rei, it allow water and oxygen to enter. This one it allows water and oxygen to enter. Then poita inonzi cotyledon. Cotyledon basa rayo riri very simple, it stores food. This one it stores what? Food. It stores what? Food. Right? Poita inonzi plumule. Plumule is this one. Plumule is this one. This is the one that develops into what? Into a shoot. Koita inonzi iyo radical. This one radical. This one it develop into root system. Then koita inonzi tester, chikanda chekunze cheche. Basa racho is to protect or covering the seed. No inonzi dicotyledonous. Anditika dicotyledonous seed, example yacho it's bean seed. Apata todzidza the internal and the external structure. Okay, let's go. Right, tenda panenonzi monocotyledon. Itaiti dicotyledon. Why? Because tinogona kuitarisa mukati nekunze, but inonzi mono because togona kutarisa kunze chete. Anditika, that's why it's monocotyledon. Tongorine one, mono it means one, then di it means two. Right, monocotyledon turikubvira like a maize seed. Ma functions tataura ndio amwe chete, hapana chakambosiyana. How do you now determine the percentage of germination? How do you calculate germination? How do you calculate percentage of germination? Can I calculate a percentage of germination? sepanapa unongoti number of germinating seeds over total number of seeds times 100. For example, iwewe waplanta ma seeds 40, ndambokupa example, waplanta ma seeds 40 anditika ndo total number ya seeds awaplanta, then ndobva kwamera 10 ndo agerminate anditika. Once iwewe ndo germinated seeds, once iwewe calculate the percentage germination, unenge wakungoti number of seeds germinated over what 10 ndo germinated up over total ichi i40 ma seeds akajarwa times ichi 100, wakutonowana chi percentage germination, wocheka 0 na 0 about this one into this one and be like a 25. So percentage germination is what? It's 25%. I think you are assisted. Okay, on our next lesson, after the test that I'm going to give you, that is going to cover the whole of biology section, we are going to start a new section in combined science, which is chemistry. So if you have your friends, your loved ones that are not yet registered for our online lesson, let them know so that they can also benefit, so that they can also start with us as we are going to start chemistry section. Okay, thank you so much. After watching this video, don't forget to like, subscribe and share to our YouTube channel. Thank you so much.